Hello students, good morning. Welcome back to a new video of Sunanda's tutorial. So in this video I will discuss regulation of glycolysis pathway. So at first recall the steps of glycolysis. It is very basic and I have already discussed in my respiration video the 10th step of your glycolysis pathway. It is common to both aerobic and anaerobic respiration. Then it occurs in your cytoplasm. So today we will discuss how it is regulated, how the steps of glycolysis is regulated. So before going to the details about regulation, first of all there are 3 checkpoints. The whole process is a 10-step reaction. 10-step process. It is a 10-step process, but there are 3 checkpoints in glycolysis pathway. One is hexokinase. So hexokinase enzyme is required in your first step to convert glucose into glucose 6 phosphate or catalyzation of phosphorylation of glucose to glucose 6 phosphate. It is the first irreversible step of the glycolysis. Accumulation of glucose 6 phosphate checks metabolism of glucose by feedback inhibition. When the amount of product is more, then the reactant itself or the reaction itself gets inhibited. In this type of inhibition, the product glucose 6 phosphate when present at its high concentration binds to the enzyme hexokinase at allosteric site as negative modulator and inhibits the catalytic activity. So the first reaction is glucose converts to glucose 6 phosphate in the presence of hexokinase. Here, the more concentration of glucose 6 phosphate inhibits the process itself because it binds to hexokinase at its allosteric site as negative modulator and inhibit the whole process. So it is the first checkpoint. Then role of phosphofructokinase, it is the checkpoint 2. And it is also an inducible allosteric enzyme. It catalyzes phosphorylation of fructose 6 phosphate to fructose 1,6 bisphosphate, which is exergonic, irreversible as well as rate limiting reaction. So before discussing anything, I want to tell you one thing. At first, you should remember what is allosteric enzyme. So when an enzyme have another binding site apart from your active site, it is called allosteric enzyme. That additional binding site can be a modulator or activator or a inhibitor. So this step here, fructose 6 phosphate is converted to fructose 1,6 phosphate, and here the enzyme is required called phosphofructokinase. It is an inducible allosteric enzyme. The most significant feature is that it is committed step. It is a committed step making phosphofructokinase catalyzed reaction as the central and most important control point. And this step is known as PFK or phosphofructokinase is regarded as the pacemaker enzyme of the glycolysis. So various regulating agents of phosphofructokinase reaction are energy charge. High level of ATP in the cell indicate that energy charge is high and glycolytic pathway should be checked to avoid any excess generation of ATP. Then high level of AMP or ADP shows energy charge is low and cell is deficient of ATP. So glycolysis should be operated to replenish ATP, and it act as positive modulator. So here ATP, in this step ATP is regulated, or ATP is act as negative modulator, but AMP or ADP is acting as positive modulator. Okay. Then citrate, an intermediate of TCA cycle or Krebs cycle, can cause feedback inhibition of phosphofructokinase. High level of citrate signify that there is adequate supply of TCA cycle intermediates for energy production. Hence, further operation of glycolytic pathway is not necessary. Then fructose 2,6 biphosphate, bisphosphate, is an allosteric activator of phosphofructokinase. If fructose 2,6 bisphosphate concentration is more, then it will activate phosphofructokinase to act. Then role of pyruvate kinase, and it is the checkpoint 3. Pyruvate kinase or PK catalyzes the conversion of phosphoenol pyruvate to pyruvate, which is the last step of your glycolysis pathway. It is the third controlling step of glycolysis. The regulation of pyruvate kinase is facilitated by the following agents: ATP, ADP and AMP, or energy charge system of cell control of the cell control the action of PK by the same mechanism as seen in PFK. When there is a lot of ATP, so there is not required requirement of further generation, so the whole process is stopped. But when there is a more concentration of AMP and ATP, the cell required to operate the glycolysis pathway. Then acetyl coenzyme A is another regulating or controlling agent of pyruvate kinase. When it is present in higher concentration, it inhibits the pyruvate kinase and pyruvate dehydrogenase. It is formed from oxidation of pyruvate in presence of pyruvate dehydrogenase, and then it enters into the Krebs cycle. High level of acetyl coenzyme A indicates that intermediates of TCA cycle are sufficient enough to complete the cycle and to generate required amount of ATP. Hence, there is no use of further utilization of pyruvate. Thus, acetyl coenzyme A inhibits pyruvate kinase by allosteric feedback inhibition. When there is more concentration of acetyl coenzyme A, it will inhibit pyruvate kinase by binding at its allosteric site. Okay. Then high level of fatty acids can also allosterically inhibit pyruvate kinase, indicating that alternate source of energy can be generated in the cell. Then fructose 1,6 bisphosphate is allosteric feed forward activator of pyruvate kinase. Once the required amount of fructose 1,6 biphosphate or bisphosphate is formed, then it drives the rest of the reaction along with pyruvate kinase reaction forward to achieve the overall goal of carbon flow and energy production. So what is feed forward activator? Sometimes the presence of a product activates or stimulates the whole reaction. So when fructose 1,6 bisphosphate is formed and present, it acts as allosteric feed forward activator of pyruvate kinase. It drives the rest of the reaction along with pyruvate kinase to carry forward the reaction and achieve the goal of carbon flow and energy production. Thus, fructose 1,6 bisphosphate is regarded as the crucial intermediate of glycolysis. Then alanine derived from pyruvate is negative modulator of pyruvate kinase. High level of alanine indicate that ATP, that is energy charge, is high, for which further ATP generation is blocked by allosteric inhibition of pyruvate kinase. So it is all about your regulation of glycolytic pathway. I hope you really enjoyed this session and get benefit from it. In my next video, I will discuss another important topic of botany. So stay tuned and please like my videos, share with your other friends and subscribe to my channel Sunanda's Tutorial. Thank you.